Welcome back. Glad you're hanging in there with me. We're going to continue from the cell division because we talked in terms of chromosomes and DNA and all that, okay? So here's one of the most important things to be known, and I think I may have done it in the first video. Is that showing up all right? DNA, is a molecule you've heard of, allows you to make a molecule called RNA, and that allows you to make proteins. And as it turns out, proteins rock the boat. They're one of the most important things on the planet. So DNA makes genes, and then there's this molecule that it has to use in order to make proteins. But if you had DNA that couldn't make proteins, the DNA would do no good. So how I look at genes and proteins, DNA, is that you may have seen at an auto mechanics place, they have these big killer tool kits that have 1,000 different tools in it, wrenches, screwdrivers, feeler gauges, and they're different sizes, different shapes, right? That would be like the different proteins of life. And the thing is, every one of those tools in the toolkit was made from a particular mold to make it. That would be like the DNA that has the template. So protein is the product and DNA has the template. So whenever you find DNA that's responsible for a particular protein, we call that a gene. Okay? So that's what a gene is. It's a specific piece of DNA that allows you to produce a specific molecule. So you start off with DNA and then you get things from it. So the first thing you get when you have DNA is this molecule called RNA. And once you get RNA, you can make protein. So when you talk in terms of genes, what you hear a lot is gene product. What's the gene product? What's the gene product? That means what is the gene making? So the first thing that the gene makes is RNA, and then after you get RNA, you can make proteins. Now as it turns out, we're not going to discuss that in this video, is that some RNA doesn't get turned into proteins. It does a significant job just being itself, okay? And there's some names for this. For instance, the process of going from DNA to RNA, what is that called? You remember? It's called transcription. Transcription. When you go from making RNA to make proteins, it's called translation. So there's certain pieces to remember. Translation, transcription. If it's transcribing, you're making RNA. If you're making proteins using RNA, it's called translation. Okay? Now, the deal is all this jazz right here, and what turns genes on, and all this stuff over here, this is a type of genetics called molecular genetics. And then remember with the chart that showed the chromosomes, that's more Mendelian genetics. Alright, so often in teaching, people will study molecular, and then we study Mendelian, and then people often don't put it together. Now, I said Mendelian. Whose name comes to mind when you think of genetics? And the word Mendelian gives it away. Gregor Mendel, an Austrian monk, who died really thinking he was a schmuck because he did his work, he wrote it up, he had it mathematically sort of complex, and the people could not appreciate it. So then years later, persons were looking at his work and they say, whoa, snap, that guy was into something. And now, if you ask, they know the name of Gregor Mendel, but he died not knowing. They took his books and work and a lot of that stuff and said, throw that out, kill the bone. So whenever you do Mendelian genetics, and I'm going to speed it up now, I know you're accustomed to it, you talk in terms of genes. And you may be accustomed to the language of dominant and recessive. And some other words, phenotype and genotype. These are in your book, and you can, see it. Genotype and phenotype. Genotype, those are your actual genes, your DNA. Phenotype is what happens because you have certain genes. You're short, you're tall, your eyes are a certain color. Okay? So now I'm going to fast forward and rely upon what you know. Genes, when you analyze them, you may do something like this. You may have seen this called a Punnett square. Punnett square. And you do something like this. One parent is like this and another parent is like that. What will their children look what you do is you take one parent, put it here, one put it right here, and then you combine them in the boxes. So this will be a big A, this will be, pull this one down first, big A, little a, little a, little a, big A, little a. So now we have these letters. So if it's the big A, let's say, equals tall, and the little a is short, and for a dominant gene, the general idea is you need one copy of it because it will dominate the other gene. That isn't exactly right, but it's okay for learning genetics. We have two big A's. So will this organism show a phenotype, what it looks like, short or tall? We look at our key. It's going to be tall. This one has a big A and a little a. What is it going to be phenotypically, short or tall? Tall. Because it has, this one is the same, and this one has two little a's. It doesn't have a tall gene, so it's going to be short. So in a very simplistic way, this is a way to approach looking at genes, even though it gets more factors into it that Mendel didn't know about. For instance, you know more about genetics than Mendel does, even before these videos. He just didn't know. He never knew about DNA and translation and transcription. And now we talk about it like, everybody knows that. Okay? So this is how genetics works. And what's interesting is you have 4 boxes. So let's see how many boxes have 2 A's, and that's called homozygous, heterozygous. Okay? You have 1, 2. So how many have 1, 2? How many have 2 of them? 1. How many have a big one and a little one? 2. And how many have 2 little ones? 1. So what that allows you to do is to talk in terms of a ratio, a phenotypic ratio of what they look like. Will this be tall or short? This is a genotypic ratio, excuse me, let me correct myself. Homozygous, heterozygous, homozygous, recessive. So it's 1 to 2 to 1. So this is a ratio. But when you look at what they look like phenotypically, these two give you 3 that are tall and 1 that's short. So that's what's called the phenotypic ratio of what it looks like. Phenotypic ratio is what it looks like. Tall, tall. 2 are tall by being heterozygous. 1, so that's 3 to 1. So genetics is pretty interesting. I like it. I would teach genetics, and you can work genetics by knowing raw numbers from organisms and figuring out the genes of the parents, or you could be given the genes of the parents and just ask you what would the children look like. So I like genetics from that standpoint. Now, in some cases, what happens is this is one gene pair. And by the way, any one of these is called an allele. Allele is an alternate form of a gene. That definition typically doesn't mean anything. What it means is if you have an allelic pair, there's one manifestation of those ATCGs that gives you what we call recessive, and then there's a variance that could be only one substitute in a long string of ATCGs, and it gives you this one. Okay, so that's what makes it weird. You could have 3,000 ATCGs, and you change one of those bad boys, you change the RNA, you change the protein, you change what happens. The person could have a devastating disease, or it could be they become some kind of big muscle superstar. Okay? So everything about you is determined by your genes, and that's pretty intriguing. So this is the skinny of genetics. And when you have problems with this, then you have problems. Okay? There are a lot of genetic disorders. Pretty much everything that happens as a disease has a genetic relationship. Okay, so this is just an introduction. Whenever you study genetics relative to humans, it's called human genetics. Okay, and then you have husbandry when you're interested in organisms. You want to have better cows and grapes and all that. Okay, and now humans have gotten so they can go in with ATCGs and say, let's change this. So you can swap genes off, change things, and that's bioengineering. Okay, where you're using genetic engineering, where you are changing things, and you get a different organism. When you are sick, you want to get some medicine or therapy, so there's the notion of gene therapy. Can you go in and fix a gene? So it came out, I don't know, 20-some years ago. It was like, we're going to save and fix everybody, and it turned out they're not like that. But in the future, it will be, especially as nanotechnology kicks in. We'll be able to do things that you never dreamt of, you see? So this is your introduction to genetics. This is one pair of genes, so it's a monohybrid cross. If I were to put in a big letter like a BB, and I'm just writing it here because of space, we have two pairs of genes, this one and this one. And if we have another pair, CC, CC, and notice I wrote two capital C's, then that is a trihybrid cross, okay? So you can have monohybrid, di, or tri. If you really understand the monohybrid, then you can, it's easier to move to the more complicated crosses, okay? Like this one has 4 boxes, and the more of these you have, the bigger your boxes get, see? But there are some interesting ways to do it. If you're interested, take in biology department, Bio 351, which is genetics. And there's a lab to go through. One of our professors teaches more molecular, down with the ATCGs and the genes and transcription, and another professor that teaches it is more Mendelian, dealing with crosses like this. Both, okay, they're the same thing. One is just, more, it's grosser, larger scale, and the other is down at molecular. So we see that you are a certain height, but when you go to molecular, you try to figure that out. So anyway, here's the brain. And your brain is made up of cells, and cells have the DNA in it, and it's important in making you who you are. So here's the brain. When you're born, you have a brain, don't you? Well, when did that occur? It started very early in development, where your heart and your different organs came into being. And the DNA caused it to be that way. So when you're in mommy, prenatal is so important, because that's when all this DNA, these genes are turning on and turning off and deciding what they're going to do. This is what makes prenatal care so important. By the time a child is 2, 3 years old, there's a lot of things that have happened that kind of lock in that person's potential to do different things, you see. And it makes it a little bit harder to try to correct it. If you get good prenatal care and, good engagement with children when they're small, then it makes it a lot easier when they get older, okay? Otherwise, it's a little bit more challenging. So that's it for genetics, and I really like it, and thanks a lot. Goodbye.